The paradox of viruses. How does our immune system respond? So, are viruses the origin of life or the bringers of death? As with most dichotomies, it is not that simple. Viruses both challenge our lives and make them possible. But let's start with the challenges. The human immune system. Antibiotics have no effect on viruses, since how can you kill something which is not even alive? Instead, we must rely on our own body and its defenses, both innate and acquired. Even vaccines require our own body to do most of the work. The first line of defense is a physical barrier, like the skin around your body or the membrane around your cells. But some viruses will inevitably slip through the cracks, like a cut in your skin or in one of the thousands of litres of air you breathe every day. Your sinuses have sticky hairs and mucus to filter out most of the solid matter, but some will always get in. Humans have perhaps the most inefficient sinuses of any animal. Due to our relatively flat faces and ability to walk upright, the drain for the mucus in our sinuses is above the mucus itself. Without the help of gravity to clear the channel, humans are more prone to nose colds and sinus infections. The common cold is caused by rhinovirus, which means nose virus. Once a virus is within the human body, including the lungs, the innate immune system will be triggered immediately. First, a complement of 30 plus proteins made in the liver are always floating freely in the bloodstream and may be activated by the viral presence. Some will sound the alarm, some will latch onto viruses and tag them for destruction, and some may even kill them directly by piercing their membranes open. When a virus breaks through a membrane and infects the first cell, the cell releases interferons. They are a group of protein molecules made by all cells which warn neighbouring cells of an impending viral infection and encourage them to prepare their defences. This includes several enzymes that interfere with and slow down viral replication. Such interferons are perhaps our greatest defense to viral infections. Single-stranded RNA viruses like coronavirus, the common cold, and the flu, however, are more difficult to distinguish from the usual cellular activity and are therefore more easily spread. Sometimes reinforcements are required. Since we are a multicellular community, also known as an animal, we have cells that are specialists at destroying viruses and other pathogens. Once the distress beacon is activated, an elite team of special forces is sent from a command post to the front line. White blood cells are the soldiers of the immune system. All blood cells originate as stem cells in the bone marrow. Most turn into red blood cells to carry oxygen, while a small fraction turn into white blood cells to fight foreign invaders. About 1% of blood volume is white blood cells, and there are thousands of them in every millionth of a litre. The vast majority of these are called neutrophils, which quickly follow chemical signals along the blood vessels, then squeeze through tissue to the front line. Along with many support and signalling functions, they can also come in guns blazing and directly swallow or otherwise dissolve foreign material. Most of this is non-living substances like dust and splinters, but they can also engulf bacteria, fungus, or human cells tagged for destruction by interferons or complement proteins. Many will die in this brave charge and leave only pus behind on the battlefield. Even these soldiers can get infected by viruses, which throws the battlefield into further chaos since they don't know who to trust. But eventually, the big artillery will lumber in and mop up. Macrophages are giant versions that can swallow any cell, even neutrophils that have engulfed foreign matter. If it is not human, they will blast it to smithereens and gobble it up. To reduce the delay, macrophages are permanently stationed in high-risk areas such as the lungs, liver, and lymph nodes. However, viruses are masters of subterfuge and can often disrupt the defensive plan. Human cells usually carry a tag that identifies them as friend, not foe, but once inside, viruses or parasitic bacteria can hide from the attention of the army. The cell has sensors on the inside and a distress beacon, but if a virus fully commandeers the cell, these features can be disabled. An arms race of defensive measures and countermeasures have been evolving since cells and viruses first coexisted. A clever trick in the cell's arsenal is the ability to show on the outside what's going on inside. MHC molecules embedded in the outer membrane display random fragments of proteins from within the cell. If it is infected by a virus, some viral fragments will be put on the outside to alert other cells of its presence. All viruses have developed some countermeasures to the immune system of their host, and the better the countermeasures, the more infectious they become. If they can block the distress signal or remove the red flags, they can develop more of a beachhead before the reinforcements and heavy artillery arrive. They interfere with the interferons. For example, the current coronavirus pandemic is more infection than the seasonal flu because it uses a more effective combination of countermeasures. It blocks protein markers on the outside of the cell, camouflages itself with certain proteins, and produces enzymes that shred antiviral messages before they can be sent. But our bodies have a whole other line of defense. After the shock troops have charged in to quell the brunt of the attack, a more sophisticated and specific response is available to eradicate the enemy. Sometimes you need to bring in intelligence officers, not just heavy artillery. Perhaps the most remarkable part of this targeted response is its ability to adapt and acquire new defenses. This chemical evolution can occur relatively quickly and does not require evolution over generations. B cells are another type of white blood cell that makes and delivers antibodies, also known as immunoglobulins, which are Y-shaped protein molecules that attach to a specific foreign substance. While there are a small and limited number of interferons and complement proteins, there are literally trillions of possible antibodies. Each human B cell will churn out thousands of different antibodies at random that are attached to the surface as receptors. If one of them connects with a specific foreign substance like a lock and key, it will trigger the release of identical antibodies and cloning of the B cell that made it. The antibody can deactivate, tag for destruction, or even destroy the foreign substance itself. A good example of an antibody is antivenom. If you were stung by an animal you have never seen before, perhaps one of the billions of antibodies currently in your body will happen to neutralize it. Similarly, some antibodies will connect to virus shells and incapacitate them. For example, the novel coronavirus breaks into cells by attaching to an ACE2 receptor on its membrane. If antibodies clog the prongs on the outside of the coronavirus, it could not bind to these receptors and could not infect more cells. This is what a vaccine might achieve. Antibodies also further activate the complement proteins, which further complement the work of the antibodies. Indeed, some complements which are always in the blood flow can only bind to viruses in the presence of a specific antibody. The innate immune system buys time for the slower antibody system to kick in. B cells can also transform into long-living memory B cells, which store any effective antibodies in case they are needed again in the future. In complicated cases, such as where a virus is already inside the cell and has blocked the distress signal, another white blood cell is enlisted to decipher the clues. T cells begin as stem cells in the bone marrow, but mature and are trained in the thymus gland above the heart, hence the designation T for thymus. If B cells are intelligence officers, then T cells are the secret agents or special forces that operate behind enemy lines. Regular T cells are experts at recognizing distress signals, red flags, and viral fragments found on the surface of cells. The tools of their trade are a huge number of random receptors on their surface, some of which may perform the required function. Due to the risk of this diversity through randomness strategy, they require further training in the thymus, and many do not make the cut. T cells have the authority to order infected cells to commit suicide, so you would not want them running wild. Another type of T cell, called killer T cells, assassinate infected cells by piercing their membrane and injecting enzymes to destroy it from the inside out. If virus countermeasures have been particularly successful, then yet another type of T cell, called natural killer T cells, come on the scene. They are experts at identifying cells with suspiciously low levels of surface markers or interferon activity, and then kill it. Such cells are particularly important in fighting this coronavirus, which are masters of disguise. The sensitivity of the immune system to irregularities must be finely calibrated. If it is too weak, you get cancer, and if it is too strong, you get autoimmune diseases, and in between lies viral infections. Other T cells, called helper T cells, mediate the process in complex and little understood ways. When macrophages come on the scene, they release chemicals which activate helper T cells, which release other chemicals to activate B cells and increase production of antibodies. These antibodies then activate killer T cells. Communication between white blood cells is mediated by a complex system of proteins called cytokines. We are barely scratching the surface of how they work. Finally, some effective T cells will transform into memory T cells, which can live a long time in dormancy and record their success for potential future use. They are why we are usually immune to reinfection by the same virus. Bacterial immunity. Until 2007, it was thought that only animals with backbones have such adaptive immune systems. In fact, many single-celled organisms have one as well. For example, many bacteria species are able to capture snippets of DNA or RNA from invading viruses and form DNA strands known as CRISPR arrays. These strands can contain over 30 different viral fragments, and the newest one is always added to the front of the chain. Some are inherited, while others are added during the lifetime of the bacteria. This allows it to remember viral encounters and more easily detect them in the future. It is a library of virus samples. If the same or a similar virus returns, the relevant segment will attach to its complement virus and make more copies of itself to attach to others. This will attract Cas9 enzymes to cut the virus genes apart. So the CRISPR-Cas9 system allows bacteria to acquire immunity to new viruses. This system is now one of the most powerful gene editing tools used by humans. But there's more. Even viruses can have immunity. A virus called ICP1 was found infecting a cholera bacteria and also has the CRISPR-Cas9 system. Since the genes are identical to its host, they were probably stolen from the bacteria. However, the resourceful virus uses this system not only for defense but also to attack. If this system is deactivated, the virus can no longer break into its host. Benefits of viruses. While viruses have caused humans a lot of grief, we may not be here at all without them. About 8%, or about 100,000 pieces of the human genome, is now composed of DNA inserted by retroviruses like HIV. These genes are only passed down to offspring when the egg or sperm cell of our ancestors were infected. There are certain viral genes found in humans, birds, and tuna fish alike, suggesting that our common ancestor was infected by a virus before our evolutionary path split almost 500 million years ago. The more closely related to another species that we are, the more viral DNA we share in common. What was first believed to be useless baggage, research now suggests that some of these viral elements still play an active role in our bodies. For example, a viral gene called HERV-H that is shared by all humans and great apes is necessary for their embryonic development. Through a little understood network of multiple switches and RNA intermediaries, it keeps stem cells pluripotent and able to turn into any type of cell. If this viral gene is blocked, the embryo development will stop. It also protects the fetus from viral infection while it's developing its own immune system. But it goes deeper than this. Evidence suggests that another viral element in the genome of our pre-mammalian common ancestor gave it the extra tools necessary to evolve a placenta and give birth to live young. Reptiles lay eggs with a completely separate supply of nutrients, but resources are wasted when the egg is not fertilized or dies before hatching. Mammals, on the other hand, continuously supply nutrients on tap, and if the fetus dies, the leak will be turned off. But this approach comes with a complication. The mother's immune system may see the baby as a foreign invader and seek to destroy it. Since viruses are experts at countermeasures to inhibit the immune response, perhaps some of their mechanisms were co-opted to allow the parasitic fetus to live inside its mother host. The placenta evolved to safeguard against this by keeping the blood supplies separate while still allowing the flow of nutrients in and wastes out. Furthermore, the human genome, as well as most primates outside of the Americas, contain another viral gene that produces a protein called syncytin. This protein is used by viruses to fuse cells together and assist in their spread between cells. In humans, however, the protein assists in binding together the placenta and uterus and building the thin yet strong barrier between the two blood supplies. So a viral gene that evolved to spread infections has been repurposed to support life. Are these viral benefits the silver lining in an otherwise abusive relationship, or are they fundamental to our very existence? Conclusions. So are viruses the origin of life or the bringers of death? Are they very complex molecules or very simple organisms? Which came first, cells or viruses? Which is more advanced? The complex genome of an amoeba with 670 billion base pairs, or the efficient genome of an influenza virus with 13,000 base pairs? Are humans really the pinnacle of evolution? How has coronavirus driven the evolution of our species, our culture, and our consciousness? I don't know. but it has been interesting to think about big questions for a change. If you have truly grasped this material, you should be left with more questions than answers. The origin of viruses is a paradox, after all. But hopefully, you have learned not to take our little companions for life, or your own body, for granted.